Mashburn here, and today I'm going to talk to you about protein synthesis. So let me see if I can get everything up and running. And why are we going to talk about protein synthesis? What does that mean? Basically, we're going to make proteins. So protein synthesis is making proteins. You've had your cell biology lectures so far, and so hopefully by this point you've kind of realized there's a lot of proteins going on in the body. Think about the cell membrane, so those membrane proteins that you use for moving things across the membrane. We have antibodies that are made out of protein. Your muscles are made out of protein. There's lots and lots of proteins in our body, and those have to be made and repaired and replenished all the time. And so we're going to talk about how we make those proteins. We need basically a blueprint for those proteins, and the blueprint is encoded for in the DNA. So your DNA is your blueprint for all the proteins that your body needs to make in order to be alive. And this DNA is divided into basically genes, and each gene is a segment of DNA or a part of the DNA, and each gene codes for a polypeptide. Codes for one polypeptide. And a polypeptide is just what? A string of amino acid chain, and when you add amino acid chains together, forms a polypeptide, and we call that a protein. We're going to talk a little bit about triplicates here in just a minute, so I'll come back to that in just a minute. Alright, so we talked about DNA replication, and that was copying the DNA before you divided the cell when you were undergoing mitosis, so a cell could divide and make more and more cells. So we've got some new things we're going to learn, new vocabulary for protein synthesis. One is transcription and one is translation. You need to make sure you know which one's which, what transcription does, what translation does, and where they occur. So transcription always occurs in the nucleus, and translation always occurs in the cytoplasm. And basically what transcription is you're basically making a copy of the message. So you're copying, kind of like a copy of the DNA or a copy of the message. So your gene, remember the gene that codes for the protein, is encoded here on this DNA. Well, you make a copy of that. So you make your copy, and this is called transcription. And the copy of the gene that you make when you transcribe is called mRNA, and the M stands for messenger. And there's a lot of details on how this actually goes along. We're doing the need-to-know basis, so don't get too caught up in the details. I just basically want you to know transcription is when you copy the gene, copy the DNA, and make a messenger RNA copy. And translation is when you actually make the protein. I'm going to talk just basically how these work. Okay? Alright, so transcription occurs in the nucleus, translation occurs in the cytoplasm. So let's start with transcription and talk about how that works, and then we'll go on with translation and talk about that process. Well, let me go, one more thing I need to talk about. When we talk about protein synthesis, we have to talk about the different types of RNA. We've talked about DNA, right? And the DNA is a double-stranded helix that it codes for your genes, it's your blueprint. Well, the RNA is kind of the workhorse. It's what we use to help us make the proteins. Okay, so they're kind of the workhorse in protein synthesis. And we have three types of RNA. We have messenger RNA, which we call mRNA, ribosomal RNA, which is called rRNA, and transfer RNA, which we call tRNA. And messenger RNA is basically the message, it's the messenger. So it takes the copy of that DNA, those instructions, carries those instructions for making the protein from the DNA to the cytoplasm. Okay, so it carries it from the DNA through the nucleus out into the cytoplasm and delivers it to the ribosome. The ribosome is where protein synthesis actually occurs. The ribosome is the site of protein synthesis. Okay? Associated with the ribosome, you have something called ribosomal RNA, and it's a structural component, a part of the ribosome that works with DNA to help translate that message So the ribosomal RNA is basically a workhorse. It helps to make the protein. And just wait till we get to the picture. It'll make a little more sense. Then you have the transfer RNA. Okay, and the transfer RNA, it's bringing the amino acids to the ribosome to build the amino acid chain. So remember, to make a protein, you have to string amino acids along to make a polypeptide. Well, each one of these amino acids has to be brought to the chain, to be added to the chain, and that's what transfer RNA does. It brings the amino acids to the ribosome so that you can build your chain. So just hang on, we'll show you the picture, and hopefully this will all make sense. So let's go back and talk transcription. What's going on in transcription? Remember, this is where we take the DNA and we're going to turn it into a message, a copy of messenger RNA. So here's your double-stranded DNA. You have an RNA polymerase. It's basically going to attach onto the DNA. It's going to separate the DNA, kind of like the DNA helicase, kind of like how that works. Separate it out. So here's your double-stranded DNA. It separates it out into two different strands of DNA. And remember how we had the adenine and the cytosine and the guanine and everything when we were making DNA? You're seeing those same things again, except now when you have what happens is your RNA polymerase, it starts to build this copy of mRNA. And instead of thymine, it adds uracil. So you still have cytosine and guanine going together, and adenine. But wherever there was a thymine before, there's now a uracil. Don't get too caught up in that, but there's no thymine in RNA, only uracil. So you have this RNA polymerase coming on, moving down the DNA, and it's building this strand of mRNA, this copy. So this is called the transcription. Don't get bogged down in initiation, elongation, and termination. That's more than I want you to know. I just want you to know that RNA polymerase makes the mRNA. Okay. So you may see this at some point. I think it's been on the T test before, so that's kind of why I throw it on here, so you'll know what the heck they're talking about. We have what's called the genetic code. And when we look at a messenger RNA, there's all those adenine, guanine, uracil, cytosines lined up. Well, every time there's 3 of them, we call that a codon. Every 3 bases is called a codon. And each set of 3 codons codes for a different amino acid. So this is an amino acid, this is an amino acid. You don't have to know the different amino acids. That's isoleucine, valine, alanine, threonine, proline. You don't have to know that. But each one of these stands for a different amino acid. And that's our genetic code. So if you see something on a T-test talking about a codon is basically how we code for an amino acid. That's how we read the DNA, read the RNA to figure out which amino acid we want to add to our polypeptide chain. Again, notice, because this is RNA, you have uracil. If this had been a triplicate code of DNA, everywhere you saw a U, there would be a T there instead of a U. Again, I don't want you to memorize which codons code for which amino acids, but several students have said there was something like this on the T's and they didn't know what it was talking about, so hopefully that kind of gives you an idea of what that genetic code is all about. Alright, so we've transcribed it. We've taken the mRNA, we've transcribed it, and now we've taken it through the nucleus and out into the cytoplasm. So here's our message, and we want to turn this message into a protein. We want to translate it. We want to read this message and translate it into something else, into a protein. So this mRNA is then bound by a ribosome. So the mRNA is released, and a ribosome attaches to it. And this ribosome can basically read these little triplets. So every time there's 3, it reads that, and it knows which corresponding amino acid it needs to add to the chain. So it's going to read each one of these 3, and it knows which amino acid it needs to add to the chain. So for example here, it's like, I have a C, C, and a G. Well, I've got to pair the opposite, C, G, G, to this site here. And so in order to do that, it's looking for a tRNA that matches that. So here's your transfer RNA, the one that looks kind of like a guitar. Associated with every tRNA is an amino acid. So the ribosome is going along, it's reading the code, and then it's bringing in the appropriate tRNA with its amino acid and adding it to the chain. That's very simplistic on how this actually works. Don't worry about A sites, don't worry about T sites, don't worry about E sites. Just know that you transcribe in the nucleus. You make a copy of the mRNA, the message, make a message of the gene. You send that message into the cytoplasm. A ribosome attaches to it. It reads that code and attaches the correct tRNA to it to build your chain. Now, there are some tutorials inside Mashburn that you can watch. It'll help you can kind of see this 3D animation and maybe clear it up a little bit for you. But don't, like I said, don't get too bogged down into the details. Now that you've made this protein, it's not ready to go. It's got to be kind of packaged and processed and matured before it's ready to be a functional, usable protein. And so that's what you do on the rough ER. So the rough endoplasmic reticulum, endoplasmic reticulum, it's kind of where you process the protein. So here's your ribosome, here's your messenger RNA, here's your ribosome, and you're building your polypeptide chain. And you notice this is associated with the rough ER. Rough means rough ER is where the ribosomes are on the endoplasmic reticulum. And as this protein goes down inside the rough ER, it's going to have a sugar group attached to it. Again, don't worry too much about that. But this is a signal for how it needs to be packaged. And once the protein is complete, it's going to be packaged inside one of these vesicles, and then it's going to go off to the Golgi apparatus. And there it's going to be fully matured, and then it's going to be released and go off to wherever it needs to go. It's going to stay in the cell, maybe go to the membrane, it's going to be an antibody, whatever it's going to be, it's going to then be a mature protein. Alright. Again, that's very simplistic view of protein synthesis. It's really all the detail I want you to know. If you have any questions, call me, email me. Let me know if you have any problems. Good luck, and I will see you again in a future video. Let me say one more thing. I forgot one more thing. Sorry about that. These proteins, once they're fully functional, just so you remember, you may see this again. These proteins, they basically have to be folded into their proper shape. So make sure you go back and look back at chemistry and remember the difference between a primary structure protein, a secondary structure protein, a tertiary structure protein, and a quaternary structure protein. Because at the end, once a protein's been made, then it has to be processed, and you're going to get those different shapes of proteins. So go back and remember what's the difference between primary, secondary, tertiary, and quaternary. All right.